Okay, Biology 107 fans, I wanted to give you a little tutorial on looking for where genes are in sequences of DNA or RNA. And here what I have for you is a sequence, a double-stranded sequence of DNA. And what I told you to look for was the sequence of ATG, right? ATG, or if you're looking in RNA, you look for AUG. And the most important part of this is that that sequence is going in the 5' to 3' direction. Both of those. Okay? So what you would do here is take a look at the top strand. You see it is going 3' to 5'. And so if you're going to start on the top strand and you're going to look for ATG, you want to go from the right-hand side here and go across, right? You go one nucleotide at a time. You say, ooh, here's an A. AT. No, that's not going to be it, so it's not that one. AT. No, not going to be that one. ATT. AC. No. All right, you can keep going. AGT. AGT. No, that's close, but not it. A, no. Blah, blah, blah. Here's an ATA. Very close. All right, not it. CAT, no, ATG. Ooh, there's an ATG. Well, that's actually a mistake, people, because I didn't know I had that in there, but this would be a coding sequence, and what you would do is you would go ATG as your first codon, and if you look at the table, you go A as your first, T is your second, G, right, ATG, encodes for methionine. So this would be a methionine, and then you would go 3 nucleotides at a time after that in the same direction. So you would have a GGC, GGC, which one is C? GGC, that would be a glycine or a G. Then you have a GAT, so you have G again, A, go down to this square, GAT is an ASP, that's a D. And in this case, you wouldn't be able to do anything more because you don't have enough nucleotides. You only have 2 nucleotides here at the end, and that means, all right, you're done with that. So if I ask you for what is the sequence of the gene, you would say to me, okay, on the top strand, I found a gene that encodes for the protein MGD. Okay, this doesn't have 5' or 3'. You don't put N-terminus and C-terminus. Okay, do not put that. It is just MGD. You can link them together if you want, and that would be the answer. Let's say there was no ATG in the top strand. Then what do? Well, in that case, what you do is you go across from 5' to 3'. Now I'm showing you in blue. You look at this first A and you say ATG? No. And then I keep going. I look, ooh, here's an ATG. So if you look at the codon chart, we know ATG is methionine. So this would be a methionine. Then your next codon would be TAT. T is tyrosine, which is a Y. CCG, which is a proline. Oops, there's my proline. TTA, is leucine. CTC, you just keep going. So you have a CTC, which happens to be another leucine. GTA, is valine. ATC, is isoleucine. TGA, TGA. Oh, look, there we have a stop. So that's a stop. The little star indicates a stop. So on the answer on an exam would be the gene encodes M, Y, P, L, L, V, I. Alright, so I think if you walk through one of these questions, you should be able to find where the gene is in any sequence of DNA. If this were RNA, the big difference would be that RNA is a single strand, and you would look, right? You would still be looking in the 5' to 3' direction for an AUG. And that's it. I hope that helps you, and I will talk to you soon.